My name is Nikos Konstantinidis. I am a postdoc in Claude Desplan's lab in NYU. And I will present to you my project, which has to do with neuronal diversity and phenotypic convergence in a nervous system. So before I start, my interests are generally about neuronal diversity. So I'm interested in understanding how neuronal diversity is being generated. And I'm interested in studying this in two different temporal scales. So I would like to understand how, for example, during the few days or months of development, some neural stem cells are able to give rise to a range of diverse neurons, both in terms of morphology or function or molecular identity. But also, I'm really interested in understanding how, during the millions of years of evolution, you can have a neuron that then by, for example, duplication of a lineage, it can duplicate itself and then generate, subfunctionalize and generate again two different neurons. And these projects are tightly linked. These two questions are linked because I think that understanding the evolution, you need to know how neurons develop. So in order to study that, I joined Claude Desplan's lab, who has a history in studying neuronal diversity. And in the lab, we're using the Drosophila optic lobe as a system to understand neural diversity. So this is the visual system of the fly. So here, this is the eye. So the eye is composed of 800 omatidia. These are unit eyes that see one point in space. They're composed of, they actually harbor a few photoreceptors, 8 photoreceptors, that then send their projections, send the information to the optic lobe. And the optic lobe is composed of these 4 neuropils that you see colored here: the lamina, the medulla, the lobula, and the lobular plate. The lamina is the first processing center. It gets information only for motion vision, motion detection, so it gets black and white information, and it forwards this information to the medulla, which is downstream. The medulla gets information from the lamina and from the color, and gets color vision information from the retina. So this is the biggest processing center in the optic lobe, and it has about 60 different cell types. Then the information is forwarded to the higher processing centers of the optic lobe, which is the lobula and the lobular plate. The lobula analyzes information that have to do with global motion vision in the 4 cardinal directions, so forward, backwards, backwards to forward, and up and down. And the lobula decodes different information, and it's much less studied than the lobular plate. The reason why we're studying this system and why I was interested in working in this system are 2. So the first one is, first of all, there is neural diversity. So this is a paper from the late 80s where Fischbach and Dietrich, they used Golgi staining to be able to visualize the different neurons of the optic lobe. And here you can see a small fraction of them, so about 20 of them, but you can see that even by morphology, they're very distinctive. They're very distinct. And of course, they don't know anything yet about their physiology, their molecular identity, and so on. So they identified about 60 different neural types in the whole optic lobe. Now we know that there are about 100. The other reason why I thought this was a good system to address my question is because Claude's lab and other labs have done a pretty good job describing the development of this neural diversity. So here, these are the neural stem cells. They're called neuroblasts in Drosophila. And what people have found is that as a neuroblast ages, it expresses a series of different transcription factors, which are color-coded here. And not only they express these transcription factors, but these transcription factors are then responsible for the generation of different neural types. So here you have a neuroblast that proliferates. It divides to self-renew itself, and then give rise to an intermediate progenitor, which will then give rise to 2 different neurons. And these 2 different neurons will be different from the neuroblast that expresses homothorax than the neurons that come from a neuroblast that expresses islets. So this way you can generate a range of different neurons. Also, this is a notch binary decision. So one of the 2 neurons will express notch, the other one will not, and this will also generate 2 different neurons. So these 2 methods, along with the origin of the neuroblasts, where exactly in the brain they come from, this gives rise to the diversity that we see, to almost all of these cell types that we see in the optic lobes. So what I wanted to understand initially was how from this initial specification, from this initial neuronal specification, these neurons then differentiate and then acquire their final morphology and their final molecular identity. And I'm still working on that, but so far what we've done is that we used as a proxy the transcriptome of the adult neurons to try to address our questions. So to get access to transcriptomic data from the adult neurons, we use two different approaches. So this is a collaboration between me and another postdoc in the lab, Katarina Caporali. And what she did was she tried to identify different lines, transgenic fly lines that express GFP in specific cell types in the optic lobe, in only one cell type in the optic lobe. And she used this to express GFP. She isolated these cell types and she performed bulk RNA-seq. And now we have information about 20 to 22 different cell types. And this is very good because we get pretty deep RNA-seq data. However, we're restricted by the number of Gulford lines that are available. So as a complementary approach, I used single cell sequencing. I used Drosophila in collaboration with Rahul Satija's group, which is here also at NYU. And we essentially sequenced almost every single cell of the optic lobe, of a fly optic lobe. Drosophila essentially is a technique that uses microfluidics to isolate one cell with a bead that carries a cell type specific barcode. And this way, all the mRNA of the cell is tagged with a specific barcode. And although we sequence all the cells together, we can then identify the cell where its transcript comes from. So essentially, you get a single cell transcriptomic, single cell information about the RNA content. This technique has an advantage that we can sequence every cell that we want, so we don't need any kind of markers. But the disadvantage is that you need to a posteriori cluster the cells based on the transcriptomic content and annotate them to identify which cell type it is, because you've lost all the morphological characters. So this is what we did. So we sequenced about 55,000 single cells, and we used several bioinformatic approaches to anyway cluster them in 52 clusters. And this is how a Disney plot looks like. So you can see that many of these clusters do pop out, but apart from that, there are many clusters that seem to be one on top of the other. So the first thing that we really wanted to do is to try to identify whether these clusters are biologically meaningful, whether they correspond to different cell types. And so we spent quite a bit of time and we tried many different things to do that. The first thing that we did was try to put them in a tree. So this is a hierarchical clustering of the clusters. So these are all the 52 clusters that we generated. And the first thing that was quite encouraging is that the first split was between glia and neurons. So glia and neurons are the more different cell types in the brain. So we're happy to see that all the glial cells clustered together and the neural cells clustered together. The second thing that we wanted to do was to compare between the RNA-seq data, the bulk RNA-seq data, and our clusters. And the first thing we did in order to do that was to select some markers coming from the clusters. So here is a heat map where each line is a gene. Each line is a gene, and that was selected as a marker. Each column is a cell, is a single cell, and they're arranged in clusters that you see here, separated by the white lines. And you can see that these are the clusters, these are the markers for cluster 1, 2, 3, and so on. And you can see that there are markers that are not expressed exclusively in one cell type, but they can differentiate the different clusters. And we use these markers to compare with the RNA-seq data. However, because the RNA-seq data were coming from a completely different approach, we decided to try to simulate single cells. So this per cell depth was much lower, obviously, than the RNA-seq data. So we tried to simulate single cells from the RNA-seq data, simulated single cells that have essentially the same transcriptomic information. So they're much shallow depth. So here you can see the simulated single cells. So each column again is a simulated single cell. They're arranged based on the RNA-seq data they came from. And each line is exactly the same markers that I showed you earlier. These are the cluster markers that come from the dropsy clusters. And initially, we tried to visually match them. So here, for example, is all the simulated single cells from cell type T1, and we tried to visually match them to one of the clusters. And we're able to match this here, and this is one of the examples. We did it for almost every cell type that we had sequenced using bulk RNA. We had the bulk RNA-seq dataset, and we're able to match them. As I told you earlier, we have about 50 clusters, whereas we know that there are about 100 different cell types. So we knew from before that there should be clusters that harbor more than one cell types. And we're relatively happy to see that the clusters that contain more than one cell type contain cell types that are pretty similar to each other. So C2 and C3 have a similar developmental history, and they look alike. Morphologically, the same for T2, T3, and the same for the lamina wide fields. We also calculated Pearson correlation, and we were able to also statistically verify that almost every cell type matched to a single cluster. Then after that, what we tried was to look at markers that we could identify from our dropsy, so some of the cluster markers that were expressed in only one or few clusters, and try to visualize where they are expressed in the brain of the fly. So we selected a marker. We used the fly Drosophila genetic tools to express GFP in the expression pattern of this specific marker. And before I go there, I want to tell you also, the other thing is that we found, we also did the same for a cell type which is called TM1. We were surprised to see that this matched to two different clusters. And actually, we were very surprised by that, because TM1 is a cell type that we didn't believe there are subtypes. There are about 800 of these. Each one of these cells corresponds to one single omatidium. So we're very surprised to see that it corresponded to two different clusters. But when we went back and restained in the brain, we saw that essentially this line that was sequenced consists of two different cell types. So TM1s expressed, so this is the medulla, the lobule, and the lobular plate, and the lamina. So TM1s are these cells here. They project through the medulla to the lobula, while T1s are also here. Their cell bodies are here, and they project to the medulla and in the lamina that you see here. So we're very happy to see that using our dropsy clusters, we were able essentially to get a line that was expressed in two cell types and divide it into its constituent cell types. And this was very encouraging. And as I told you earlier, we also used we identified some markers, and we looked where they're expressed in the brain. So this is a gene that was supposed to be expressed only in one cluster, and we see it expressed in only one cell type that we can identify, and the same for this one. So we're able to give a name to many of these using FACS-sorted data and the different markers that we identified. And this is where we are currently. So we're able to give a name to all the glial clusters based on the literature. So many of these have been sequenced before, so we're able to give them a name, and we're able to give a name to some, but not all of the neurons. But given our controls, we were now confident that we were looking at something that were meaningful clusters of cells that were similar and come from the same cell type. So then we decided to address our questions. So we wanted to see how neurons acquire their characters. And we're particularly interested in transcription factors. So we wanted essentially to be able to connect characters, specific traits with specific transcription factors. But before we do that, we wanted essentially to differentiate between two different models. So assuming that we have two different neural types, neural type 1 and neural type 2, that are completely different, but they express a glutamate, so they're both glutamatergic. We wanted to separate between two different models. So the two different models are either you have a global regulator of genes that endow glutamatergic identity on the neurons. So in this case, you have the red transcription factor that drives the expression of V-glut in both neural types. Or you could imagine a model where you have neural type specific transcription factors. So you have a blue transcription factor for one cell and a green transcription factors for another cell. And that's what we wanted to differentiate. So we decided to generate a machine learning model, a random forest model, that would connect the transcription factor expression of a cell type to its gene expression, the non-transcription factor gene expression. I would call these effector genes from now on. So what we did is that we took our 52 clusters, we separated them in a training set and a test set. We trained the model using the training set, and then we gave the model the transcription factor expression of the test set, of the clusters of the test set, and we asked what is the gene expression in these clusters, what is the non-transcription factor gene expression. And that's what you're seeing here. So here you're seeing gene expression of every single genes of the 13 clusters of this test set, the actual gene expression versus the predicted gene expression. And our model performs pretty well in predicting gene expression. In neuronal clusters, it does a little bit more poorly in glial cells, and we believe that the reason for that is because we have fewer glial clusters. The model is not being trained very well, but we focused then on the neuronal clusters. And what we wanted to actually find is, if we have a gene of interest, a specific gene of interest, can we find, using the exact same model, the transcription factor that is responsible for generating, for the expression of this gene. And that's what we did. We focused initially on neurotransmitter identity. So in the optic lobe, we have 4 different neurotransmitters. We have cholinergic, glutamatergic, GABAergic neurons, and we have 2 dopaminergic neuronal types. So I selected genes that are expressed exclusively in these neurons. So this is the choline acetyltransferase, the vesicular glutamate transporter, the vesicular monoaminergic transporter, and an enzyme which is responsible for the synthesis of GABA. And based on the model that I showed you earlier, I predicted I identified the transcription factor which is better correlated with this gene. And we used RNA-seq to test it. So this is a wild-type brain. So this is the lobula, the lobular plate, the medulla, and this is the expression of CHAT, choline acetyltransferase, in the brain. So upon RNA-seq against the transcription factor that was predicted to regulate CHAT, you can see that there is a strong decrease in the expression of CHAT in the brain. So this was very encouraging, but what also came out of this experiment is that although in some layers of the neuropils, you can see almost complete elimination, a very strong elimination of the expression of CHAT, in some others it appeared to be almost unaffected. So this pointed to us that there might be some other transcription factor that's regulating the expression of CHAT in other cell types. And this also in combination with another observation that we made, which is that this is a heat map of the transcription factor expression. This is the tree that I showed you earlier. Red are glia, green are neurons. So we could see that there are some paneuronal TFs that are expressed almost in every neuron. But then we saw that most of the other transcription factors are expressed highly in only one cell type. And this is not 100%, but there was a very strong trend towards that. So that actually was the first hint that we had that there might be some phenotypic convergence in the brain. And what I mean by that, so if we imagine that these ABC are 3 cell types that are present in the brain of a fly, and the bold line indicates a specific trait that they share. So we can think of 3 different scenarios how we got there. So one of them is that their common and neuronal ancestor had this trait, and this trait might be regulated. In this case, the most possible is that it's regulated by the same transcription factor, this orange transcription factor. The other scenario is that they acquired this trait independently, in which case they might also use the same transcription factor if they were primed to use this transcription factor, perhaps by the presence of some binding sites upstream of the gene, or they might have used different transcription factors to achieve the expression of this gene or this trait. And we cannot really compare, we cannot really differentiate between these two, but we can differentiate between these two and this one. So if we have 3 cells, cell A, B, and C, these cells here, we can expect that the gene, one could be either regulated by the same transcription factor, so they might have a common activator, or they might have a neuronal type specific activator. And this allows us to make some predictions that we can test. So one prediction that we can test is that if this is the case, we should be able to identify an almost one-to-one correspondence between this gene and this transcription factor in our data set. And the contrary would be true for that. So we shouldn't be able. And that's what we tried to do. So we took all 9,000 non-transcription factor genes that are expressed in the brain, and we tried to correlate them with a transcription factor. And what we found is that about 3,000 of these genes we could correlate with a single transcription factor, and about 6,000 were correlated with, could not be correlated. So they supported a neuronal type specific model. However, when we looked closer, we saw that these genes were essentially expressed in 1 to 2 clusters only. So they're essentially expressed in very few clusters, and that's why they correspond one-to-one with a specific transcription factor, whereas these are more broadly distributed, so they're expressed more or less in more or less clusters. And the other thing that was, so this also showed us that there is this phenotypic convergence that we postulated. And also what we found is that most of the genes that are important for neuronal development, axonal projection, neurotransmitter synthesis, are genes that support also this neuronal type specific activator model. And this brings me to, yes, so this essentially supports the model too. So we believe that in the Drosophila optic lobes, most of the genes are regulated by different transcription factors in a cell type specific manner. And this has been shown already to be the case also in the worms. So in C. elegans, the lab of Oliver Hobert has shown that this is also the case for genes that are important for neurotransmitter synthesis, and this is actually where this figure comes from. But what we showed that this is a more general phenomenon, and it's not only restricted to neurotransmitters. And we believe that since this seems to be the case in both flies and worms, there's a good chance that this will be found also to be the case in more complex neuronal systems. So this brings me to my conclusion slide. So what I showed you here is that we have used Drosophila to identify cell types and specific markers for these cell types. We used various different methods to quality control our data. So we used FACS-sorted cell type specific transcriptomes. We used glial transcriptomes that were available in the literature. And we used known and newly identified markers to annotate some of our clusters. And we annotated all our glial clusters and some of the neuronal clusters. We're able to generate a machine learning model to predict transcription factors that regulate terminal genes and hence some traits. In the case I showed you here, there were neurotransmitter identity. And we were able to show that there is phenotypic convergence in the optic lobe, which means that there are cell type specific transcription factors that regulate terminal neuronal characters. So with this, I would like to just acknowledge Katarina. So this is a work that we did together with Katarina and of course Claude. We did everything in his lab. And Rahul Satija, who helped a lot with the Drosophila, and Lou and Shaima, and all of the Desplan lab members and the Drosophila community. And I would like to thank Dennis for organizing this and all of you for listening.